Okay, so we're very delighted, very pleased to have with us today Professor Amir Amedi, who is a professor in the School of Psychology in the Interdisciplinary Center, IDC, Herzliya in Israel. Amir did his PhD studies in computational neuroscience at the Hebrew University of Jerusalem under the supervision of Professor Udi Zohari, and also in collaboration with Professor Rafi Malach from Weizmann. At that stage, he already started to investigate how somatosensory and auditory inputs affect visual cortex activation in normally sighted individuals. And then he moved to investigating also congenitally blind individuals. He then continued to a postdoc with Professor Pasquale Leone at Harvard and NIH. After that, he returned and joined the Department of Medical Neurobiology and Cognitive Science at the Hebrew University as faculty. At 2019, he joined the IDC and he founded, and he's actually the founding director of the Baruch Ivtzer Institute for Brain, Cognition, and Technology, and the Ruth and Meyer Rosenthal Brain Imaging Center that includes their new MRI facility. The institute he founded and his lab study the human brain, its interface with technology, science, and application of human rehabilitation in various conditions like visual and auditory impairments, as well as emotion regulation. He's also an adjunct professor at the Sorbonne University at the Institut de la Vision in Paris. Amir Amedi is one of the founding members of the International Multisensory Research Forum known as the IMRF and chaired its annual meeting in Jerusalem in 2013 with participants from all over the world. He published over 100 papers and also book chapters and has over 10,000 citations. He won dozens of prizes for his work, including the Kirill Prize from the Wolf Foundation, Career Development Award from the International Human Frontier Science Program, and others. He was recently elected as one of the Genius 100 Visionaries that includes 16 Nobel laureates, as well as additional exciting visionaries. Professor Amir Amedi also holds patents in the field of multisensory integration and rehabilitation. His work has been featured in high-profile scientific journals as Nature and Science, and also in distinguished newspapers and journals as the Washington Post, National Geographic, Wired, and others. His lab is consistently funded by the prestigious ERC grants. And without further ado, I'd like to welcome you, Amir, to our seminar to tell us more about your exciting research.
Thank you, Sharon. It's a big pleasure. I don't know if it's probably an extra special fun to be here and kind of nostalgia. Sharon and I did overlap during our PhD with Rafi Malach, and even put a couple of slides from that period. But I'm going to focus today on how experience shaped the human brain, nature versus nurture factors. And I'm going to especially focus on a new work with Professor Serge Dumlin, Dr. Shir Hofstadter, that look into the emergence of tonotopic maps and a full 2D soundscape representation of sounds in the visual system and across the brain in the absence of visual experience. This is a work that we recently published in Neurobit. Before, as a little intro, but I think you did a wonderful work to highlight the new center. Basically, we tried to bridge neuroscience, computer science, rehab, engineering, medicine, and psychophysics and physics. So people that are in the lab coming from very diverse background. I'm going to mainly talk about current work with Dr. Shir Hofstadter and Dr. Kasia Siesla, and also, and this is the collaborator, Stan Dihan, worked in collaboration with Stan Dihan, Serge Dumlin, and the previous PhDs in the lab, especially work I did with Elas Raymond. I think I will skip my background and just to say that the new facility that we are having, and I'm mentioning it also because of two things. One, I'm very excited from this new multidisciplinary approach in which we can reprogram sound in a 3D sphere in a room with about 100 speakers We integrate virtual reality, cave technology, virtual reality technology, haptics technology that are all can also be in the scanner. So I won't focus too much on this work today, but I did want to mention it. And we are also collecting, in addition to MRI signals, physiology signals, and hormonal signals in collaboration with the lab of Dr. Ruth Feldman. And of course, we use all the various technologies. And basically, the vision of the lab is to develop new technologies, use them to study the brain in a new, unique way, and combine them with up-to-date technologies, and then use these insights also to go back and improve the technologies. But the main focus is basic science, as I'm going to highlight here. And as I mentioned, I'm going to feature mainly work with, in terms of collaboration, Laurent Cohen, Joseph Sael, and Stanislas Dihan, also known as my Paris gang. And then 3 hours train ride to Amsterdam, the director of the Spinoza Center in the Netherlands, Serge Dumlin, which together with Brian Wondell, developed the population receptive field mapping. I'm going to highlight in the main part, the second part of the talk. Yeah, I'm going to, I think, skip this, and just first of all, for those of you who will be bored by the long lecture, I'm going to give the bottom line in a lay language version, and also it's kind of I try to be a bit provocative. But I will try to provide here evidence to suggest that the brain is actually not organized according to the senses. It's not an input-based, sensory-based operator. And this separation to visual brain, auditory brain, and so on is wrong, and it's only an illusion to a large extent. But rather, the brain is organized by computations that are serving specific goals. And usually you will have one sense that is more dominant to perform a given task, but this is flexible if you have the right technology, or if the brain find like bypasses. So this is kind of some revised model of brain organization. I then want to highlight the fact that a lot of the things that we think about as visual, for example, reading, are actually in fact rely heavily on other senses. So actually reading is a multisensory phenomena in which we took something that had enough time in evolution to develop, like language, and have specialized brain area that are auditory in essence to communicate, and then embed 5,000 to 10,000 years ago this information in vision. And this created a big revolution, because then you can keep it for generation and so on and so forth. So even something that is we think about as vision and visual word form area and so on is a multisensory process actually. So this is important. And also if you think about it, I love to argue that, and when I decided what I want to focus my research on, it was vision in the interaction with the other senses, because actually if you think about it, almost all the important things that are important for us in life are multisensory. As I said, in the central part of the talk, I would like to highlight a new study, which basically take inspiration from the classical work of Marigan Casseur. Of course, I will show a few slides, but just as a general lay language description, what Marigan Casseur did in MIT, he asked what happened if I'm going to, in the ferret, which is system after birth is still not fully tuned to the senses, he rerouted the visual nerve with a very complex surgery and operation into the auditory cortex, and asked whether retinotopic maps can emerge even though this cortex was not, was not programmed in evolution to carry out vision, and definitely not retinotopic maps. So we asked the following question. One, is this possible in humans? Two, can this happen reversely? So can we have tonotopic maps in the visual system? And three, can this happen in the adult brain? So I will try to, I'm going to present to you some very intriguing results about this using a very interesting technology that take visual information and embed them in sound. And this way we could do retinotopic experiment in people that never had visual experience, which sounds like an impossible thing to do, yeah? Based on this setup and this study and many other studies, I'm going to argue that we should retire to some extent, and this is where I'm a bit provocative, that the concept of critical windows, critical periods of critical windows, which suggests that we have a start, we have an end, and that it's closed to a large extent, should be retired, because and we should retire it with reversible plasticity gradients. So basically the idea is that yes, the brain is more plastic when we are young and it's decreasing in its plasticity capabilities. But in contrast to the concept of critical window, this is reversible. And if we can reprogram in the age of 50 an adult brain in the visual or in occipitotemporal or occipitoparietal or in retinotopic areas to have new tonotopic maps, then we should rethink about all this concept of plasticity gradient. Of course, this joins to many other labs that show in various models like cataract, congenital cataract, like congenital blindness, congenital deafness, and so on and so forth. This is not going to be an overview lecture, so I'm not going to give the review that we recently published, Heimler, with Dr. Benedetta Heimler on this topic, but I'm going to mention it. And we can leverage this neuroplasticity to develop new technologies and so on and so forth. So I think in this audience, I don't need to mention the seminal work of Jubel and Wiesel, which were the first to record neurons from V1, and also were key in suggesting that you must get visual experience during development in order to have all the key specializations like the two pathways, like the ocular dominance, category selectivity, and so on and so forth. Now, if you think about it, this concept basically limits to some extent how much we can reprogram a brain that is 50 or 60 or 40, or a brain that had stroke, and so on and so forth. So what I'm going to ask here in the first part of the talk is what underlines the emergence of anatomically consistent specialization we have in the human brain. Yeah. So for example, we started the work in LOC that was discovered by Professor Rafi Malach. And then you have the basically specialization. So first of all, you have specialization for ventral stream and dorsal stream. And here is a zoom in into the specialization within the visual system. In the ventral stream, similarly, we have specialization in dorsal stream. We have several work on navigation and on motion and on localization that Dr. Elena that is here, and Dr. Shachar Meindenbaum, and so on and so forth, study navigation and other functions, but I'm going to focus on the ventral stream. And then on top of these specializations that are very consistent across individuals, we have these topographic areas that are considered to be this type of topographic maps that we have in the tonotopic areas, in the auditory system, and the retinotopic and the visual system. These are the areas that's supposed to be the most dependent on visual experience. So this would be the focus on the second part of the talk. So as Sharon mentioned, we shared some time in Rafi Malach's lab, and for my first work during the PhD, I showed that the LOC complex is as active when sighted people palpate objects, and it's dropping in the signal in both the visual system and the somatosensory system when we are doing visual recognition of textures or of scrambled images. Now, to summarize a few studies that are summarized here below and are already published, we show that visual and tactile object selectivity happen in the ventral visual stream, but there is no response for auditory objects at all. And this peak activity in LOC happens even with zero visual experience. Okay, so our suggestion at the time was that it's not about what is shared with vision and touch and is not shared with audition, is that both of them can decipher the exact geometrical shape. So we suggest that the ventral stream, and especially LOC, is actually a geometric shape operator, and it's most easy to do it and most accurate in vision, but it also can be done in touch. So you actually don't need visual experience to show this selectivity. So, so to summarize this, it's only an illusion that the LOC in the ventral stream is visual, just because vision is the most efficient sense to compute this geometry computation. Now, the critics, or the follow-up work, was that actually we'll palpate objects very early during development. So, okay, we don't need visual experience, but this still, the brain is super plastic in the infancy and the first years. So maybe this can only happen if you palpated object and extracted geometry when you're a kid. And the fact that the auditory objects, like the typical sound of the hammer and so on and so forth, is not activating this LOC, is because usually sounds for us are associative. So if God, for example, or whoever, the spaghetti monster, or whoever you want to believe is switching typical sounds of dogs and cats, you don't get any information about the shape, but after, by statistic learning, you learn that meow meow is a dog in front of the door. But there are some animals that develop in evolution this amazing ability to decipher geometry, very exact 3D organization of objects and objects in the surrounding, like bats and dolphins. And there is the amazing work of Yossi Yovel and Nachul Ulanovsky. Dolphins can also do it. So the question that we ask in this project, in the first ERC project that we had, what if we can teach blind adults at the age of 20, 40, 60 to extract shapes from sounds, just like we bats and dolphins do. But this time they don't take the tens of thousands of years of evolution to develop this ability, but we teach it over the course of several months in about 100 hours of training. So this is a very ambitious project and very difficult. And basically it's following the footsteps of Paul Bacharita, Dr. Paul Bacharita, which basically had this genius idea to say, why don't we help blind people by putting, in the beginning, he had this monstrous dental chair with all these actuators on the back, and when we have the letter V or a line going down, like in V1 or up or different orientations, then the actuators vibrate on the back, and in a few hours of 10 of hours, they're able to do it. Now, when I started to think about this model and combine it in humans and with brain imaging, I was thinking that the auditory system is much more relevant and much more interesting because it's had an order of magnitude more information, and it's also very easy to do MRI experiments with it. So in the lab, we use two types of technology, one developed by Peter Mayer from the Netherlands, which is grayscale representation of an image. And we also develop our own in-house system, which is called the iMusic, which adds also the color. And we know that most of the receptors in the eye are color cones, especially the high resolution part of the brain. And that color is key in segregating object from background and so on and so forth. So we developed this technology that enable you to decode color, but also the shape, the geometry, the location, and even the brightness of the environment. So a blind here that is looking at these apples would have to create in his mind eye this low resolution, 2000 pixels, between 1000 and 2000 pixels image. By the way, 2000 is roughly what you need to face recognition. And if he's able to create this image in his mind eye, he can know where is the red apple, and since he knows the location, he would grab it. So this sounds a bit science fiction, but again, as I said, the part of this talk is nostalgia, nostalgia, sorry. I like to have this quote from Rafi Malach that always said that we are seeing with our brain, not less than we see with our eyes. And a very nice examples to it is this FedEx logo. I guess most of the vision scientists know this, but for those of you who are less from this background, I can, what happened to me was that I looked on this logo many, many times. And one day, after I already saw it hundreds of times, I noticed that between the E and the X, there is a white arrow. And what is interesting, so is that it's enough to tell it to you one time, and now from now on, every time you see a FedEx trap, this arrow will jump on you. So it changed completely the vision. So this shows it's not just bottom up, it's also top down. So the idea in sensory substitution is that if the eyes or all the visual pathway is not working, why don't we access this occipital cortex by the ear? We have all the auditory pathways. We would process that as sound, but then later, since we are seeing with the brain and not with the eyes, or we seeing also with the brain and not just with the visual pathway and the retina, then we will be able to see this image. So this was the vision of Paul Bacharita. I won't have time to, one of the questions I have is how can we teach someone in 10 hours or 100 hours to see with sounds? So I don't have enough time, but I'm going to do a very quick demo. And just since it's Zoom, if you feel like opening your cameras and do it together with me, it will be really nice. So I can see you. But basically, the idea is that in the algorithm that black pixels are quiet, and as it's more and more white, the volume increase. Higher notes are high in the pentatonic scale, and lower pixels are low notes in the pentatonic scale. Why pentatonic? That's the topic for a different talk. And basically, a diagonal line that might be represented in V1 would sound like this. Okay, so now for the little demo. Now let's try to do together, for those of you who are feeling like doing one demo together, the rest so open your camera so also I can see you. Let's try to do together, you don't need to open your sound, but let's try to draw together a smile now. I explained to you 1 minute the principles of this, yeah? So let's start with, I will give you a note. T, sing with me. I want to see your lips moving, T. And now let's sing together a smile. Okay, so the sound would start going down, yeah? T. Okay? Now let's try to draw a sad face, yeah? So T, T. Okay? And now finally, an indifferent face would be T. Okay? So it was nice to see all your faces. Now back. So basically, this is lesson number 1. So lesson number 1 goes around stimuli that are very similar to what V1 is doing. And as the training progresses, they are doing more and more difficult. So for example, up-down lines or thin and thick lines. Let's do it again. Yeah. And after like 10 hours, we can already start teaching like symbolic faces. Yeah. Okay, I cannot play it. So if the eyes, by the way, are the 2 high notes. Yeah. Okay. Now, we don't have time to go. And by the way we do color is by programming timbre that can switch very quickly between pixel and pixel. It sounds easy, but it's actually a very complex technologically. Now, let me show you a few things that blind can do after 10 to 100 hours of training with this. So basically, we train them to be bats and dolphins. I'm going to play a 1-minute movie, and then I'm going to explain several things about it. So in the first movie, you are going to see someone learning to read the word Abba. Of course, this was more not to replace Braille. For this, we developed a different technology that uses a combination of Morse and Braille. but it's a great to teach using letters. They are very, it's like the next step after line orientation.
רגע, האמצעית זה ב'.
And you have the English translation here.
א', אבא יצא חד.
אלה לא רק אותיות, על פי הטכנולוגיה עיוורים יוכלו להרכיב מערכת ממוחשבת שתכלול מצלמה פשוטה, שהתמונות שהיא תקלוט יעברו תרגום לצלילים. שימו לב לסרטון הזה, איך האישה הזו סיגלית, עיוורת מלידה, מצליחה למצוא בין שלל החפצים שלה בבית. זוג נעליים.
Okay, so I'm going to maybe I will show you one more movie of the use of colors and facial expressions.
מעולה. עכשיו. מעולה.
Okay, and here you remember the example of the apple, the red apple. This is actually, all the people that you saw so far in the movie are actually congenitally blind, very carefully checked by neuro-ophthalmologists that verify this, and there is ways to verify this. And what you see here is actually a live demo in front of 10,000 people of Hamutal, one of the blind subjects that we had that was willing to do it and share her experience, do exactly what I told you with the red apple. And then we are going to do the same with Coke and Sprite and shuffle them online in front of the stage. So just to show you that you can do color and you can do location, and you can combine it with the body scheme and reference frame. This is actually a different case, and this is actually the first retinal implant patient that did an Argus 2 surgery and was able, so he was seeing all of his life. I'm not going to show brain imaging work with him, but only from congenitally blind because of the question of can we develop specialization without visual experience. But what is amazing with him is that he can compare, he contacted us after he saw some media coverage on it, and he came to learn the method of seeing with the eye music because he felt that the Argus 2 didn't give him enough resolution. It's only was 60 pixels, and he could not do. It was amazing for him to see light after a decade of darkness, and he can use it to some stuff, but mostly it doesn't give him shape recognition. And it was very difficult. Took him. One thing that people don't realize about retinal implants is that just like sensory substitution, it takes about several months to 1 year to be able to see even very simple things. Actually, in 2 weeks, we can achieve much more than what we achieve with a retinal implant after a year. And we did this comparison with people like this patient. But what is also really nice is that he described the phenology, the conscious experience he have in learning to see with sound.
כשאני חושב על צורה וצבע וכולי, התמונה עולה לי במהירות מאוד מאוד גבוהה. אין דבר כזה שאני אזהה משהו ולא אראה אותו.
So actually describe how after some time when he picked up a banana, he suddenly have an hallucination of the actual banana image with the color and with everything jump into his mind eye. Now let's switch gears and look at the brain. And this is a very nice image that shows that in congenitally blind, and this is a group of over, we now have data from over 10 congenitally blinds that show that while we take the visual images and encode this information in 2D soundscapes, in sounds that represent using the pitch and the time, a 2D image, the brain is doing the inverse transformation. One thing that I do want to mention here, it's actually very important after these very cool movies, is that this is the resolution of the vision, but not only in space, not only in terms of the pixel resolution, but also in time. It takes them in the beginning several seconds to generate the image in front of them. So of course, if the images are moving, like somebody is approaching you or there is a car, you will be lost, okay? You can't do it. So there is some limitation to these methods. Now we try to combine touch with the sound, but that's not the topic of the talk. Now, the advantage of having them a few seconds to create the image is that we can actually see the neurochronometry, how the information flow in the brain from the onset of the stimulus until they manage to read the word or recognize the face or the object and so on and so forth, or the facial expression and so on and so forth. So what is really cool in this movie is that you see how the information comes from the ear, from the camera transformed into the ear using the technology, and then it travels in the auditory system, activate the temporal cortex, and then activates the ventral stream all the way to V1. Now, what I'm going to show you in the second part of the talk is that actually, when we are doing a retinotopic mapping using the PRF method on the soundscape, on the two axes, and one axis is tonotopy and the other axis is time, for the tonotopy axis, we get receptive a tonotopic map, a mirror symmetry tonotopic map in the auditory cortex for the soundscapes. While for the rest of the brain, we get these tonotopic maps also before training, but after training with the super user, we get tonotopic maps only outside the auditory cortex, in addition to the A1 map and the RT map, in various locations in the brain. And this would be the focus on the second part of the talk. But I do want to dedicate 5 more minutes to a line of basically over 30 papers we published over the last decade, showing that any specialization in the visual system can emerge after these several weeks of training or months in this adult brain. And all the papers that I mentioned here are a group of about 7 to 10 congenitally blind individuals. And for example, we replicated the classic work of Ungeleider and Mishkin of the what and where or how pathway, a la Goodell. And we showed that when they analyze the soundscape, even with no visual experience, when they do a shape task, they recruit the what stream. And when they do a location task, they recruit the dorsal stream. And then we went one area after the other: LOC, reading, faces, houses, navigation, and the body, and so on and so forth. I do want to mention 2 things very, very quickly. We study, first of all, LOC, of course. You remember the question about the bats and dolphins. If we are doing speeded up evolution and we teach them to analyze complex geometry with sounds, and again, the y-axis here, you see a spectrogram. The y-axis is tonotopy, is different tones, and the x-axis is time. Then we see that when we are teaching the sounds and playing and just telling the subjects, this weird sound is a horse, and we don't tell them about sensory substitution and it contains shapes, we just ask them to learn them, there is 0 activity in LOC, nothing significant. In congenitally blind, in late blind, and in sighted. On the other hand, when we teach them to extract shape, both the sighted and the congenitally blind and the late blind starting to have significant activation, and this differential activation between object and scramble, just like Rafi found for LOC for sighted in the classic PNAS paper. Okay. Now the only example I want to give you, because before I'm jumping into this V1-like representation we found using the population receptive field method, I do want to show you one example, and this is the visual exostriate body area that was discovered by Nancy Kenwisher's lab and Downing, and showed very nicely that we have a special category of analyzing the visual representation of the body. Just maybe we'll do one more demo, and I will ask you to point with your hand for 1 or 2 if you think we have now the Euro Cup, the European Championship in soccer or in football, as we call it. And so in the spirit of it, I created this demo that showed that usually we think that all the information is in the faces. Now try to think who to guess and point. If you think that this guy scored a goal, point 1 with your finger. And if you think this guy scored a goal, point 2 with your finger. So just by analyzing their faces, the expression when they scored a goal, try to guess who scored the goal and who missed the goal, okay? So who think this guy scored the goal? Do 1 in your finger, okay? So I'm seeing here about half doing 1, and if you think the other guy scored the goal, do 2. So it's about half-half. Actually, I lied to you. Both of them scored the goal, but this is the only lie I hope I had in this talk, yeah? And if you have the body information, it's very easy to know that this guy scored, and this guy's actually missed the goal from the hand and the body expression. And all of this is done in the exercise right body area, and then this information is being sent into the mirror system or to this bimodal system that have both visual and motor neurons, and it's very well kept in evolution. I like to give this very cute movies to show that even kittens can look visually and then imitate in the same movements. And it's so preserved in evolution that, you know, even humans can do it. Okay, and maybe dogs can do it, but let's keep it. Now, this is actually a very interesting question. What about congenitally blind? Because they never saw visually, but if we are now using this ability to see with the ears, give them a 2D visual image, can they imitate it? And either is Hamutal again, one of our favorite subjects.
ואני שומעת קווים אופקים, אז אני מניחה שהוא עומד ככה.
And now we scan the brain of sighted using doing objects versus other categories or objects intersubject consistency. And we see this very nice right EBA. The white pixels, if you can see them, are actually 100% overlap. So you see this beautiful overlap. But now we are doing the same thing for congenitally blind. So you see some A1 activity in the temporal, you see here gyrus, but you also see that the most significant and consistent area in the brain for analyzing these bodies is in the right EBA. And actually, if you do the overlap, if with all the subjects cited both on the group level and individual level, you cannot distinguish basically if it's a sighted doing it by vision or a blind doing it by sounds. And we repeated recently this also for faces. The reduction, of course, maybe is because we have this inversion effect that we are very good at analyzing upright faces, but when we are rotating them, the differences between two images disappear. That's a classic illusion. And basically, to summarize, our data suggests that visual cortical specialization do not need visual experience to develop. And that can happen in the adult brain if we are able to teach to extract the same information from a different sensory modality. I'm not going to show you all the other studies that we did, but basically we replicated it to all of these categories. And now comes the very interesting question, is what about the topographical gradients in the brain, the tonotopy for different pitches, for retinotopy, and so on and so forth? Can they also emerge in the adult brain? And here I'm going to show you just a proof of concept, one study that as I mentioned, drew from the classic work of Marigan Kasser. And basically, he used this very interesting model, the ferrets, and the reason is that they are very immature when they are neonatals and can be manipulated. So onset of hearing tasks take place several weeks after birth, and they have they are very intelligent and can learn to carry work. And as I said, Professor Sir from MIT developed this very complex surgery happening after birth, but very inside the critical window period, that rerouted the visual input into the auditory system. So you see here the classic from the retina, LGN, visual cortex, and here you see the pathway. They had to do a lot of surgical interventions in order for this to happen, but the bottom line is that they rerouted the information into the auditory system and showed that the rewired A1 show very similar maps to what Amir Amedi, for example, showed in V1 for different orientation selectivity. Just like the sounds that I gave you, going up, going down, smiling, and so on and so forth, okay? And this is, of course, the resolution was, and that's very interesting, the resolution was lower, and also the receptive field sizes in way A1 were larger. We are actually basically replicating some of these conceptual findings using our model. So the question, so what's going on here? He suggested the visual input during development is actually instructed to establish a specific cortical secrets in A1, which thereby exhibit functional properties similar and organization principles similar to what is seen in V1. And to form this map is not an intrinsic property of the cortex. A cortical area can come to support different maps, topographic maps. So this was a very unique example of the emergence of topographic map that is not what the evolution and the development during the critical periods or the critical window or during when the brain still develops a form, but he showed that you can develop a full, although lower resolution and higher receptive fields in the brain. And then we asked the following question. Can we do the opposite? Can we create tonotopic maps that will be formed in the visual cortex? And of course, the brain in general, because no one showed the emergence of such topographic maps for senses that can emerge in the adult brain. So this is one question. So can we do the opposite of what Marigan-Kasser did, rerouting visual input into the auditory cortex and show that topographic maps, retinotopic maps, or ocular, dominance maps can emerge in the auditory cortex. The second question we asked, can this happen in the adult brain and not during development? Can we take someone in the age of 40, 50 and show that topographic maps can emerge? Can this happen even using non-invasive procedures? Because part of the work of Marigan-Kasser showed that if you are not doing surgery and some cutting, I don't want to get into the details, but you need not every time you reroute the visual the visual pathway into the auditory cortex, you will have this retinotopic or orientation selectivity maps. And finally, can we show this in humans? Because this was shown in ferrets. So to do this, we teamed up with Serge Dumlin that created this very clever method to map retinotopy. Basically, the idea is that each point in the visual cortex have a different population receptive field. So the column is representing the properties of single neurons because they have similar properties in different columns. And then you can also map the location because each time, just like Euban and Wiesel, a line will go into the visual location. While you have fixation, it will be stimulated. And when you are entering the fovea, you have this back of the brain, the pole. And as you go more and more anterior, more peripheral, it will be, the map will be more and more anterior. So this is one dimension. And the second dimension is basically the polar angle. And this way you have a full 2D map that represents the full 2D image that fall on the retina, or that represents the visual field. What we did was very simple. We took a congenitally blind, the best user, because this was a proof of concept study. So the one that have the most experience and they can do the most impressive task. We took these visual images and transformed them into soundscapes. So usually you have this polar angle and eccentricity to map basically you think about it as a coordinate system to represent 2D. And here in the soundscape, to remind you, we also have a representation of 2D of this image, but this time the y-axis is frequency, is tonotopic, basically, organization, and the x-axis is time and stereo panning. So it will sound something like this. And then we will simulate a different part of the visual field, so-called, using the soundscapes, and then a different location. And basically, we replicated the classic retinotopic experiment, but this time using 2D soundscapes. Okay, and now let's look at the results. So basically, this is the computational method that we use that can tell you for each voxel in the brain, what is the location. Up means high pitch, low means low pitch, early in time means left side, in the middle of the time, it means that it's in the middle, and later in time, it means in the right. And also the size of the receptive field, because basically, if you are stimulated only in this specific location, it means you have a very narrow 2D receptive field. And if it's a very large receptive field, then each time you get anywhere near this location in the world, you will get activity in a given voxel in the brain. So basically, this is the model that fits the signal in each voxel in the brain independently, and also the size of the receptive field. And using this, we found this, first of all, this is brain areas, and this is stunning map to my, sorry that I'm still excited from this, but this map shows where we have a big variance in the brain. So it means that we can explain a lot of the signals using either the frequency, the time, or their combination. So basically to map the receptive fields in the brain. So this is basically how much variance is explained by the population receptive field method. And you see here there is robust activation in the auditory cortex, but also in the visual cortex, okay? Both in retinotopic areas all the way to occipitotemporal here and occipitoparietal. All of this known to be high order retinotopic areas, and also areas which you have coordinate transformation of the retinal information. Now, in addition, we can now look on each axis separately, just like I showed you before, that in vision, we can now separate the eccentricity maps showing the center, the fovea periphery, and the basically polar angle map. So we can analyze each axis separately. We're doing the same by analyzing the tonotopic axis versus the time axis, which together, the spatiotemporal signal over 2-3 seconds create a 2D image in the blind, and they can do all these cool things like knowing what is your facial expression, or pick up their shoe or the apple, and so on and so forth, okay? So now let's look at these images. Now we are adding the basically tuning of the neurons in the auditory cortex, and you see this beautiful classic mirror image map in A1 and RT cortex. So basically, using this method, we can also do tonotopy, of course, because, and by the way, we did run on this subject also tonotopy before the study. And we showed that you have tonotopic maps in auditory cortex, but it doesn't have any tonotopic maps. And this was replicated in several groups in the world that show that you get some response to sounds, but you don't have tonotopic maps outside the cortex to these subjects or to any congenitally blind. But now let's look at the rest of the brain, not just in the temporal cortex. Let's look at the visual cortex and the occipitoparietal and occipitotemporal cortex, and you see these beautiful tonotopic maps that are emerging in the adult brain. So basically what you see here is the mirror image of what Marigan-Kasser showed, but it's in the adult brain. It's the opposite modalities, so tonotopic maps in visual cortex. It's non-invasively. It did take, this subject had more than a year, even 2 years of working with this tonotopic and time devices that represent vision, but it's quite stunning, I think. So to summarize, neuropopulation are tuned to auditory frequency, both in auditory cortex, occipital and parietal cortex. These neural population are organized in multiple topographic maps, just like the auditory cortex have multiple topographic maps, and the visual cortex, just like Marigan-Kasser, show multiple topographic maps in the auditory cortex to visual stimuli if he's doing this surgery. What was really also very interesting is that another collaborator of Serge, his former student, showed that there is time maps in the brain. And very interestingly, we are seeing this beautiful mapping also to the time axis. And in one structure, we even, actually in several structures, but one was the most intriguing in areas that are around, here you have V6 and close by areas. You see here a full 2D representation, or not full, but in low resolution and with higher receptive field sizes, just like Marigan-Kasser. But in the same cortex, you have both the y-axis, the frequency, and the time axis. So together, it's very similar to the organization of primary visual cortex that we have in V1, but also in V2, V3, V6, and so on and so forth. So we have neuron populations that are tuned to auditory frequency, both in auditory cortex, occipital, parietal cortex, but not before training, only after training in the adult brain. So there is no new tonotopic maps in the blind without actually training with these sensory motor contingencies of a 2D geometry map, a soundscape map that is translated into an image and then fitting it into your body scheme and interacting with it. So I think I'm going to stop here. We have some very interesting future direction in which we are now trying to train sighted people to analyze infrared information or ultrasound information and see whether you need to have blindness to develop this topographic map, so you can do them also if you have intact vision and audition, and many other projects, but I'm out of my time, I think, and I will stop here. Another project I think which would be very interesting to this community is that we actually now have several papers showing that sighted people can learn to create a full 360 representation, low resolution in the back, high resolution in the front, and they can learn to grab objects in the back and locations, just like the blind are doing. And we are now trying to push it in terms of their, of course, it's very difficult to do it with sighted because they're not interested really to learn this topic, less than blind. But we are planning now to do longitudinal studies in which we will scan and look whether you have new retinotopic maps for eccentricity, for polar angle in the back, and see how they interact. The classic maps that were developed a la Hubel and Wiesel in the critical periods, and the new maps that are learned in the adulthood, even without blindness. So this is the setup we have. This is the new Brain Imaging Center, some collaborators, and I want to thank you here and say that if you are interested in this topic, we have several vacancies for postdocs, PhDs, and even neuroradiologists and so on in this new Brain Imaging Center. So feel free to drop me a line at amedi@idc.ac.il. Okay, thank you very much. If you have any questions, I'm more than happy to answer them.
Thank you very much, Amir. And I want to invite everybody to unmute yourself, and let's give Amir a big applause for a very fascinating, overwhelming talk. And yes, definitely, if people have questions, you're more than welcome to unmute yourself and ask. And of course, my questions will also.
I have a question.
Sure.
Hi, Amir. Thank you very much for the great talk. I was wondering if did you try to measure how many details in a horizontal plane the subjects can detect? So you were showing something like 2, I think. So yeah. This is like 2 contours. So what is the maximal ability to
actually, we did back in the day 2 studies on this, and basically we the best way to do it, I think, if I understand correctly your question, Yossi, and again, thanks for the invitation to you and to Sharon. It's a big pleasure and big honor to be in this amazing series that you organized. So basically, you're asking what is the visual acuity that we have in each axis. And
it's not only the acuity, it's acuity is another thing, is how many levels of sounds you can have. So it is, yes, it is acuity in the...
Basically, it's acuity in each dimension. So we have 2 studies, one with Elastra Amit and one with Dr. Shelly Levitzedek, that now have a lab with Be'er Sheva. And we showed one that you can have acuity that is close to the acuity that is the definition of legal blindness. So you can actually have can get quite accurate using this method. Of course, I want to mention that this was in optimal conditions. So one object. Once you have several objects, it's become a very difficult task. Very, very difficult task. So just to mention, again, this is not regular vision, slow resolution, slow. A lot of details is also to visual, to have a search with a complex scene, but it's even worse here. And the second work was actually really cool. We showed that adding color, like we did in the iMusic, improved the resolution even further. Okay, so you can actually get even beyond this by adding color information to segregate the, for example, smell and stimuli.
Yeah, and maybe this relates to another question that I wanted to address. You just mentioned it, but why is it pentatonic? I know that you are a musician,
yeah.
So maybe you like John Coltrane, but why is
so
it?
I'm actually a failed musician. Yeah, I went to the music academy to be a jazz musician, a saxophone player, and I love the pentatonic scale. It's the basis for blues, for jazz, and so on and so for rock, by the way. But what is really nice when you practice on this is that, listen, if you have just one sound, it really doesn't matter what will be the scale on the y-axis. It can be any scale. It can be major, minor, whole tone, and so on. The problem is that you need something, you don't know what will be the stimuli in front of you. So you need something that even 15 sounds at the same time will sound okay. And this is a characteristic of the pentatonic scale. Try it in the piano, and you can choose any 2, 3, 4, 5, 7 notes on the pentatonic. It sounds great harmonically.
Yeah. So it was for the pleasance of the sound.
Yes. And of course, I think the pleasance of the sound is also means how much you are going to work and practice on very difficult cognitive tasks while you are using it. So you will reach higher resolutions. Yeah.
Yeah. Okay. Thank you. Thank you.
It's a pleasure.
Can I ask a question? Hi, Amir.
Of course.
Thanks for the great talk. There seems to be a large asymmetry between the x-axis, which is time, and the y-axis, which goes in parallel. So this should put some limitation on the kind of vision you can get, because you need to do something in parallel and something serial.
True.
And I wonder if you investigated this. I mean, in terms of what you can do in visual search, or so the visual substitute.
Yeah, absolutely. First of all, it means that your vision is very slow. When we start, it takes them about 5 seconds to understand one image. So, of course, in movement or moving things, it's terrible, yeah, because your time resolution is every 5 seconds.
Of course, I'm not talking about moving, but just even static.
Yeah, but even static, yeah, it will take you 5. The best subjects, like the one I showed you, can do it in 400 milliseconds. But still, it's very slow compared to vision, or 500 milliseconds. Now, it also means that there is a very interesting interaction between pitch and time, and also you can have, like in the algorithm, stereo panning, and this type of interaction is very interesting. We never really studied them, and I would be happy to talk with you about it. This is one of the goals of this talk series, you know.
Okay. Just a quick another question is, did you ever look at eye movement during this process?
Not really, you know, it's something that we wanted to do for a very long time. And I think, as I mentioned, Dr. Elena Agius-Vela in the lab is very interested in this type of questions. And we do have some studies which, for instance, we look at the combination of the motor system and the body representation with navigation using this and so on. Eye movement would be super interesting, especially in congenitally blind. There is challenges because they have nystagmus and they.
Right.
But it will be intriguing. I think there is some very interesting question about this, and it's also important. I mentioned a little bit my collaboration, long-time collaboration with Dr. José Sahel, the director of the Vision Institute in Paris. But there is huge challenges by coupling and decoupling in prosthesis, the eye movement and the image. So it's how much you can modify this interaction and so on is a big question and very interesting. So some prostheses are actually moving together with the eyes. Some you have the eyes moving, but the image staying, and the head moving, it's moving the image, not the eyes. There is a lot of very interesting rehab questions. But again, for this community, I try to focus the talk on the neuroscience and on the relevance to, you know, critical periods and representation of the brain.
Thank you.
Amir, there are questions in the chat that I can read to you. But before that, I just want to say that Professor Sahel is actually going to be giving a talk in a few weeks here in the seminar as well.
Don't miss it.
Yeah. So one question was, would this be relevant? I mean, this is Joanne asking, for those diagnosed with cerebral or cortical visual impairment. I mean, your results?
In principle, yes, because what is nice about this approach is that for the prosthesis, you need to tailor a solution to each condition. Yeah. So the Argo still works for RP, but it doesn't work for anything else, and so on and so forth. And in principle, I think besides the very, very, very rare huge stroke in the visual system, that even there, I think it might be that other areas will take over if you do it like very extensively. It can fit to any condition, late blind, congenitally blind. The reason doesn't change too much, and so on and so forth. As long as you have intact audio or touch, we have now touch technology that Dr. Cassiesla showed you can do the opposite. So you can decode speech in touch. And now we look at the brain mechanisms of it and how do you decode sounds from vibrations on the body. We also playing with representing vision together with audition on touch. So lots to do.
Thank you. And another question by Aliran. Have you asked if the patients dream their audiovisual experience?
Wow, what a beautiful question. And this is why we are recruiting more people. I will say that we didn't do, we have a new postdoc in the lab, Dr. Amber Maimon, that I mentioned that is here in waving, that is coming from this phenomenology field and philosophy field to combine with neuroscience and sensory substitution, which we are going to ask exactly this type of question. Amber, maybe this is the first project, the second after having a paper on this retinal implant patient that we didn't ask him about dreaming, but he is mentioning, and I think I mentioned it very briefly during the talk, that as he was training more and more with the eye music, he suddenly started to have hallucinations. And he described there in some of this video how when he see a banana, he have an image of a yellow banana in his mind eye. So my guess is that yes, they will start dreaming in soundscapes. But if it's enough, if they use it enough, of course, another thing to mention, right now we can only use it in the lab. Of course, if some blind take it and get on the street and get over, with a car, my career, his life is over and my career is over. So we are not doing it. But I guess they will use it enough hours, or maybe very early in the experience when everything is so new and intriguing, maybe they would start dreaming about it. As I said, Amber, add it to the list. I think it's a wonderful question. And
added it.
Great. Another question by Gadi Bloomberg.
By the way, just to mention one more thing, now there are groups that are even using MRI to decode dreams. So in principle, we can let the blind doing it, and try to see this. Actually, this PRF method is very good for decoding, because if you have all the pixels and you train enough the decoder, in principle, you can reconstruct the image. So you can even see the images of what he's dreaming on in the scanner. But this is like these projects are super difficult. It takes a year to collect data from it. So this will be 10 years, but it will be an amazing project in itself.
Okay, lovely. Another question from the chat by Gadi Bloomrosen. Do people who compose music have very active vision areas? Do they represent music, notes, melodies, or tunes as shapes? I assume, I don't know, I assume there's big variance, but...
Yeah, we didn't, what I can tell is that we had hundreds of people over the years not reaching this level of this super user, but hundreds of people that came to the lab, and we do track their music abilities and so on and so forth. And it's a universal thing. one person couldn't do the transformation. Now, another thing I can say that we scanned both expert musicians and non-musicians with these tonotopic tasks, and none of them have topographic maps in the visual system. Okay, so to the level, and also not congenitally blind. So to this level, this is the one case studies and only case which shows topographic map in the adult brain that is topographic and so on. I don't know what happened in synesthetic that sounds trigger complex shapes. If anyone, I don't think anyone did retinotopy and tried to map this and see what's going on. Another fascinating project. Maybe this should be a collaboration with Liad Mudrik that I saw is here. I see that also Peter Mayer in the Netherlands, maybe he can comment. He developed the voice system that we used a lot in the lab. And he has written a beautiful review on phenomenology with seeing with sound. So yeah, lots of open questions on this.
I have a question.
Sure.
Sure.
Great talk, super cool stuff. Do you have any evidence that we need to worry about sort of disintegration of auditory cues for hearing? Like when these trained patients hear jazz or something, do they have little visual hallucinations or something?
No, very, very interesting question, but it seems like the just like in vision, yeah, or in multisensory, the setting and the environment determine how much would you how would you experience things, yeah? So you know that if this is in the context of this device, it's representing vision, and when I'm hearing the environmental sound, it's not. Now, it might be that if they use it like on a daily basis, then sometimes they would hear a concert and they would start seeing, you know, some shape. They would say, oh, this reminds me of a smile, yeah? I don't know, I'm just, but again, we never studied this. Again, in our heart, we are developing the technology and using this to study more basic neuroscience questions. So what happened when you have visual experience, when you don't? We study it and so on, like what happened to the specializations. But now when the team is increasing and we start to have like multidisciplinary people coming, we can start asking also this type of more phenomenology or behavioral questions.
If I can ask another quick question. So in thinking about taking this more to a basic neuroscience level and seeing if it can be done in animals, I guess I was wondering during the training, is active use by the patient necessary, or is it enough to show them the associations, this sound is a horse, and then that's enough to change the circuits?
I would put my money, but I don't have a hardcore data on it, that you would need the interaction. But it will be very interesting. What is a very interesting question if there is some people that work with animal models. My lab is not working with them, so I cannot do the studies. But like to put a camera on and this correlation to a chip after the animal was born, I think would rewire complete, like it will be Marigan Castor on steroids. I think it's and I think like moving it to the practical domain again. So this is from the neuroscience domain. Also, like if you are a blind kid and you will using this non-invasive technology and the age of 4, 5, 6, again, ethics is tough, rightfully so. Although the potential is great here. So this is another thing to study. But we are actually moving away from this to start looking on novel sensory experiences of people that have intact senses and related questions. So I think I would love to see this type of work being done somewhere in the world.
Awesome, thank you.
A pleasure.
I see I am muted, and there's one more thing in the chat. For experienced patients, have you tried to speed up the time for the audio input. Okay, this is again Eliran asking. I guess it's whether.
Yeah, actually, this is what we do. We start with a very slow, like brain imagers, time to repetition. Yeah, so it will be like every 3 seconds or 5 seconds, and then the time to repetition become faster and faster.
Mm-hmm. Wonderful, Amir. This was a wonderful talk.
With pleasure.
Yes, our pleasure, definitely. So again, I want to ask everybody to give a big thank you to Amir for the wonderful talk. And of course, I assume if you have more questions, you can contact Amir also directly. We have even the email within the talk. And next week we have here, we will have again in this very different, or at 3 p.m., the same time as today, Professor Patrick Kavanagh, who will give again a talk. So I will publicize that, but I hope you will be able to join us. We have a few more fascinating talks before we take the August break. So thanks again, Amir.
Pleasure.
And thank you. And thanks everybody for joining us.